Hello, everyone. Good morning. My name is Sarah Malanga, and I'm sitting here in Malawi. And I would like to welcome you to our second session of our workshop, Rangeland in Balance, Managing Wildlife and Vegetation. I can see in the chat we have Kelvin Kirkman. Kelvin, if you don't mind, can you please unmute and say hi and let us know why you're so much interested in today's topic?
Good afternoon. Yes, Kevin Kirkman from University of KwaZulu-Natal in Pietermaritzburg. I'm a grassland scientist, and so the topic is really interesting to me, and yeah, keen to see what comes out of it.
Thank you. Enjoy, and I'm telling you, you'll really enjoy it because, yeah, we have good speakers.
Thank you.
And I can see we have Laura. Laura, if you don't mind, can you say hi to the people and let us know where you're joining us from?
Yes, of course. Hello, everyone. My name is Laura. I work for Dancheche, Zimbabwe. It's an NGO that works with the communities, and I would like to learn more how communities can be involved in conservation projects. That's my main goal with participating in this workshop. Yes, thank you.
Thank you so much, Laura. It's good to have you here. All right, so it's exactly 12 o'clock. Oh my God, 1:12. We are going to get into it. But before we get into it, I would like to kindly ask those who joined us yesterday that we sent out a survey. And if you don't mind, please give us some feedback. We are looking forward to seeing your feedback on yesterday's session. And today, after this session as well, we're going to send out another link for today. And please don't get tired of us. Give us some feedback. Would like to improve. All right. So I know we are feeling so energized after yesterday's session, after diving deep into game capture and translocation. We talked about dynamics, hands-on work of managing wildlife population, which is just one half of the rangeland management equation. And today we are moving to the other half, which is equally critical, the landscape itself. We are getting into vegetation surveying for rangeland management. Think of it this way. If our last session was managing actors on the stage, today is about understanding and managing the stage itself, where the dancing is happening, the acting is happening. How are we going to decorate it? We will explore how to map habitats, assess carrying capacity, and how we use tools like remote sensing and quadrant sampling to make informed decisions. This isn't just about identifying plants. It's about seeing the bigger picture. It's about understanding the health of an entire ecosystem and making sure it can support a healthy population of wildlife for years to come. This is the foundation knowledge that everyone needs, that every conservationist needs to make informed decisions. So let's get into it. And now I'm going to hand over to my colleague, Kirsten, who is our host today. Kirsten, welcome. The screen is yours.
Thank you, Dr. Stella Malanga. It's really good to be here again, and I'm looking forward to our audience enjoying today's session. So here we go. Our session today is titled Reading the Land, Vegetation Assessment for Rangeland Management. And we have, as before, we have three amazing speakers today. We've got Dr. Tony Swemmer, Nyeko Mashaba, and Sachin Dursami. So moving on. I'd like to first introduce Dr. Tony Swemmer, and his talk is titled Vegetation Assessment for Rangeland Management. So, Dr. Tony Swemmer, a man who knows the savannah like most of us know our favorite coffee shop. As head of SAN's Ndlovu node since 2007, Tony has led groundbreaking long-term ecological research on everything from the impact of elephants on trees to restoring grasslands in both rangelands and mountainous areas. We'll learn from Tony how to understand the purpose of vegetation assessments, to apply core principles of vegetation assessment, including sampling, key variables, trends, functional grouping, and adaptive management, and to understand common measures of abundance, sampling designs, functional groups, and grass species identification. We will be able to interpret vegetation data using indexes and trend analysis to inform rangeland management decisions. So please join me in welcoming Tony, a true visionary in savanna ecology, ecosystem restoration, and environmental engagement. Thank you, Tony. The floor is all yours.
Hi, morning. Thanks for that very elaborate introduction. Thanks. Good morning, everyone. So I work for SAON, as Kirsten mentioned, the South African Environmental Observation Network, which is a facility of the National Research Foundation, which falls under the Department of Science and Technology and Innovation in South Africa. So we essentially a facility for doing long-term environmental research, and we run a number of sites where we facilitate research by scientists and students. And the branch of SAON that I work at is in the northeast of the country, close to the Kruger National Park. And so we do a lot of work in savannas, also a bit on grasslands. And so I'm just going to speak to you this morning on what I know of vegetation assessments. So I think yesterday you did animals, and normally the vegetation stuff is considered quite boring, especially by students compared to the animal stuff. But of course, vegetation is critical. It's the food for the animals. And it's critical to understand what's going on with vegetation, whether you're a farmer or a manager of a nature reserve or some other protected area. Okay, so I'm going to briefly go over that, the purpose of vegetation assessments, and then speak about some general principles that should be applied whenever you're doing vegetation assessments, and then a bit on the methods. And the methods, there's a lot, and it's complicated, so I'm really just going to give you an overview of some of the common methods out there. And this includes doing some basic mapping at the start, and then the field sampling, focusing on what variables you might measure, and sampling designs, and a little bit about grass identification. Okay, so to begin with, we've set this first slide up just as a little Mentimeter question. So just to get some feedback from you guys in the audience, what do you think vegetation assessments are? So you should see now on your screen that a little Mentimeter link has popped up. And if you can join that, then you'll be able to give your say here. So I've asked a question, what is the purpose of vegetation assessments? And the options are A, to maximize forage production, so the production of food for your animals, whether you're a farmer or a conservationist, or B, to determine the carrying capacity, how many animals should you have in a certain reserve, C, to prevent degradation, D, to conserve biodiversity, E, to comply with the law. In other words, it might be a law that you have to do vegetation assessment, or F, all of the above. So just take some time to answer that. Don't have to think about it too much, just whatever you think is the most appropriate. We'll just give everyone about 30 seconds or so to do that. So let's carry on. So as you can see on the screen, most people chose all of the above, and I think that is the best answer. You do vegetation assessments for a variety of reasons, depending on who you work for and where you're based. So it could be for any of those, and often it's for all of those. Complying with the law, it's not necessarily a requirement. If you say work in a protected area, you don't have to do vegetation assessments necessarily, but you are obliged to show that you're looking after the land. And so you need some way of doing that. And so vegetation assessment is normally the way that's done. And of course, it's often required as well for rehabilitation, to show that it's been done, for example, by mines, or when you need to comply with some specific requirement. For example, if animals are going to be introduced, the organization selling them to you might want to see that the vegetation is in a certain condition. Okay, thanks. Let's move on. All right, so there's a variety of reasons for doing vegetation assessments. So how do we actually do them? How do we do vegetation assessments? So before you start, you have to understand why you're doing them in the first place. You can't just rush out there and collect data and hope to do something with it. So for any reserve or farm or game reserve or game farm, there should be management objectives, and those would then lead to monitoring objectives. So you want to monitor whatever is relevant to achieving whatever you're trying to manage, and that then gives you your method. So for example, if you're a game farm and you want to maximize forage production for your prize sable, then your monitoring objective might be to monitor the abundance of palatable grasses for them. And then the appropriate method for that might be a step point or line transect. On the other hand, say you're working in a big protected area, a national park, the management objective might simply be to conserve biodiversity. And then you might just want to monitor species diversity, make sure that all your species are always present. And then you would use a different method, such as sampling quadrats or even an inventory. All right, so once you've decided exactly what you need to monitor, there's a couple of principles that can be applied, and these really apply to all kinds of vegetation assessment, whether you're a farmer or a game farmer or working in a protected area. So the first is sampling and replication. It's impossible to measure everything on an entire reserve. It's just too big, it would take too long. So you have to only measure parts of it, you sample. You measure selected things in selected areas. An important part of that principle is replication as well. How do you know how many samples is enough? You have to take repeated measurements of the same things so that when you look at the data, you can work out if you've got enough to adequately describe what's going on. So your samples are kind of pieces in a puzzle, and you have to have enough pieces to actually see what the whole picture is without building the whole puzzle. Okay, and related to that is a concept of homogeneous vegetation units. Which is basically the same thing at a larger scale. You need to sample across your landscape evenly so you get all your different vegetation types included. The third principle is about trends. One point in time, knowing your condition of your vegetation at one point in time is useful, but it's far more useful when you're looking at how it changes over time. And then the fourth principle there is functional grouping. So in most systems, you have so many species that it's really impossible to try and understand what's going on if you're trying to analyze all of them. So we group them into species which behave or function in similar ways. And the last one is adaptive management. So this is the idea of doing something with the data once you've done your vegetation assessment. It needs to be done in a way that it can inform management, that it can be used to implement new management actions if necessary. Okay, so just about the sampling. So if ecosystems were simple things with just one species and they all grew evenly spaced out across the landscape, it would be the easiest thing in the world to do vegetation assessment. So that little cartoon picture on the left shows that. And if you wanted to do an assessment there, all you'd have to do is measure one of those plants. And because they're all the same, you can then extrapolate that the whole system has a certain characteristic. But obviously that's not how the real world works. So the picture on the right is a more realistic example. We've got different species, different types of species, and they're patchy. There's a clump of tall grasses in the mid there. There's some alien prickly pear thing on the right. There's some flowering plants in the foreground of that photo. So you need to sample in a way that you capture all of those without actually having to measure the entire landscape. Alongside this, at a larger scale, when you're not just sampling a patch, you're sampling a whole reserve, say, it's useful to divide the land up into units, into blocks that are similar. So for example, in this picture, we've got a river, and then there's some riparian vegetation alongside the river, and then there's some low-lying areas beyond that, and then there's a slope, and it goes up to a plateau. So all those areas are going to have differences in soil, they're going to have differences in water holding capacity. There's going to be different vegetation on them. That vegetation is going to respond differently to things like grazing intensity or drought or fire or whatever. So normally you would group them into homogeneous vegetation units. So here I've just drawn in, for example, a lowlands, a slope, and an uplands, which is a very common way to divide up the landscape. Okay, then the principle of time trends. Okay, so let's do this as a question as well. Let's say you go out and do a vegetation assessment. There's some sort of index you create, it doesn't matter what it is, but let's say you get that value. It's about 80 out of 100. That's your result. Is that good or bad? If you go out and do a felt condition assessment wherever you're working and you get a value of 80, is that good or bad? The correct answer to that one is you can't actually tell. Because you could have a scenario like this, and here I've added in hypothetical values from before. So you'd been monitoring for 5 years, and you see that your condition is improving steadily every year. Then yes, definitely that's good. It's got up to a good value. It's improving. Things are good. But what if the situation was this? What if you had 4 years of 100, and now you were down to 80? So 80 is still okay, but definitely there's a problem. Suddenly things are going down. So then you should actually implement some sort of action or do some further investigation. So that's just to illustrate you can't really make many conclusions or conclude anything with confidence if you've just got 1 year. You really need multiple years. And especially when you're doing vegetation, because it can change very slowly. So sometimes you really need more than a few years, maybe even a decade, before you can actually see what's going on in the long term. All right, so the next principle is functional grouping. So let me just make a little disclaimer here. I have been using an AI for some of these photographs, these images. I think you could probably tell from this horrendous one. So here I wanted an image of different grass types in a savanna landscape. So this is courtesy of Google's one, what's it called, Gemini. So anyway, what we're showing here, there's different types of grasses. The zebra's feeding on short grasses. Those are obviously grasses that are palatable, that it likes. And in the background, we've got these really tall bunch grasses that are probably not very palatable. So if you were wanting to assess the vegetation and how good it is for grazers like zebra, it would make sense to group all the short grasses that are palatable, provide good forage, and all the ones that don't. Now that's really just the concept of functional grouping. It's applied in many different ways. The final principle then is of adaptive management. So you might have seen or heard of this before, but it's essentially this idea of a circular process where you have your management objectives, and those lead to monitoring and sampling. And then based on the results of that, after you analyze that, you then adjust or adapt your management. And then you go back and monitor again. So it goes on and on. So it's just to make the point, this is not a one-way process. You don't set up your objectives and then monitor and then go home. You have to do those final steps of analysis and then adjustment. The little red arrow is just showing that if your monitoring results or your assessment results show that everything's okay, then you would just skip the adapt and change and go back to sampling again the next year, or whatever frequency you're doing your assessments at. Okay, let's move on to some of the details, the actual methods of how you do vegetation assessments on the ground. So step one is mapping to get your homogeneous vegetation units. Then you've got to decide what variables you're actually going to measure. You've got to have some sort of design about how you're going to sample within your vegetation units. Related to variables is deciding what functional groups you're going to measure. And alongside that is how to actually identify the species within those various functional groups. And then lastly, indices and data analysis. So what to do with the data when you have all the raw data. So I'm going to focus mainly on sort of more traditional rangeland monitoring for this. I think most of our audience are TUT students who are doing a diploma in nature conservation. So these are, I'll speak more to the type of things they might be doing. But obviously vegetation assessments are very broad, and they are used in a wide variety of ecosystems and vegetation types, and there's a very wide variety of methods. So I'm not going to go through all of them just for the sake of time. Before I get stuck in, why not just save yourself all the trouble of all this detail and just sort of walk through the veld or an ecosystem and just make a visual assessment. So you could find yourself an expert, someone who's been working in the business for a long time, and just ask them, to walk through or fly over or drive through and make an assessment. And in fact, that's how reserve management and farm management was done for a long time. It was only about 100 years ago, maybe, that rangeland monitoring was started to be done in a more scientific way. But what are the issues of using an expert? Why don't we just use expert opinion? So we're going to go back into Mentimeter for this, and you've got 4 options here. So the first one, that there's no way to establish who an expert is. So it might be a problem if you want to use an expert in that you don't know how to identify an expert and whether they really are an expert or not. Your second option is that your expert might turn out to be biased and you wouldn't know. They might think your felt condition is in bad condition just because they work next door and they don't want your property to be as good as that one, or something like that. The third option is that you can't use an expert year after year because eventually they're going to get old or retire or move or something, and then it's hard to establish trends if you're switching from one expert to another, or if you lose your expert. And the fourth option is that human memory is not exact. So I certainly know this from my own experience. There's sites where we've been doing sampling for 15 years, and I kind of remember what it was like in the beginning, and I know there were some good years and bad years, but I can't remember exactly when or how good or how bad things were. So yeah, what do you think is the most important reason? All right, so it seems like for the majority, the possible problem of bias is the biggest concern. And after that, human memory is not exact is the second most common answer. And then some people worried about not being able to use the same expert, and no one really too worried about how to establish who an expert is. Okay, that's quite interesting. So all these reasons are valid. These are all 4, problems with using expert opinion, and why we rather use an objective scientific method. If you have someone who's using a standardized method that's been published in scientific literature, it means it's been sort of scrutinized by other scientists, and it also means that they've shown they can do it. And so that kind of establishes them as a suitable person or team to do your vegetation assessment. So using a scientific approach gets around that first problem. It also gets around the problem of bias because, all humans are biased to some degree or another. And when you're using a scientific method, it's kind of designed to remove that bias as far as possible, because you're doing things in an objective way. And once it's recorded, you can't change it. And also, if the method's established and it's recorded, then it really doesn't matter who does it. If the person who's been doing it for 10 years disappears and someone else takes over, as long as they follow the same procedure, then your data and your trend analysis should be okay. And of course, that takes care of the problems with human memory as well, because when you're doing a formal method, things are written down, they're recorded, and so they're there, they don't change as your memory changes. Okay, back to the slides. Okay, so another point about exactly what methods to use is that you have to tailor your methods according to the ecosystem you're working in. So I mentioned that rangeland monitoring was, typically done for grasslands and then savannas. Situations like in the top left there. So a lot of the original sort of methods came from pure grasslands where there were no trees, so they only deal with measuring grasses. When you're working in savannas, like the top middle photo, then you have some trees and you have to have a different method to deal with the trees. In some cases, like the top right, you can have really dense trees and short trees or shrubs. So you might need a different method to measure tall trees versus shrubs and short trees. And then, of course, in situations in drier ecosystems, like the bottom right, that's a Kalahari ecosystem, you can have very sparse plants. So your grass method that might work well, say in the Drakensberg, where there's dense cover of grasses, is not going to work so well in the Kalahari, where you've actually got a lot of bare ground and things are spaced out. And then, of course, there's other ecosystems that where grass is not the dominant plant. So the bottom middle picture there is a picture of the Karoo, where it's mostly little shrubs. And so you'd need a different method that's better at capturing the diversity of shrubs and grasses. And even the fynbos, which is the bottom left, even that sometimes requires vegetation assessment. And there again, you're going to need a different method. And these are just common biomes from South Africa. For those from other parts of Africa, again, you've got different systems and you're going to need to modify methods. Okay, but one step that's kind of universal is getting a large view of the area you're monitoring before you start and dividing it into homogeneous vegetation units. You can, if it's a small area, you could do that just by driving or walking through the area, if you know the vegetation really well. Generally, it's better to get a bird's eye view from above. In the past, aerial photography was used. So there would be high resolution aerial photographs, which are very useful, although they generally were in black and white and not that readily available and quite expensive to get. These days, fortunately, we have a lot of satellite imagery of high resolution that's freely available. So Google Earth, if you don't know it, it's a great tool for doing this sort of thing. It makes available some really high resolution satellite imagery for the whole world, and it's updated fairly recently. So for example, that picture on the right is an area just near the Palabora Gate of the Kruger National Park. This was taken from Google Earth. I sourced it just this morning. And you can see on there, there's areas of different vegetation. In the bottom left, there's a little river going through where it's darker green. So that would be a riparian zone. And then there's the lighter green patches of little dots. Those are uplands or crests where you have more sandy soil and certain types of grasses and trees. Those little dots, incidentally, are termite mounds. So that gives you an idea of how the resolution of the imagery. And then in between the lighter green areas are the sort of brownish, reddish brown areas, and these are slopes, lowlands with a more clay-rich soil. Okay, so mapping is normally fairly easy. You can do it in a very detailed way where you actually go sample the vegetation and look at different communities and use statistics to define different communities and then map based on that. But for normal vegetation assessment, that level of detail is not necessary. It can be just done from an image. Okay, then you've got to actually measure how much there is of the species or the functional group that you're particularly interested in. And this is not as simple as it seems. So for example, here's another little question. There's the picture on the left, which has got 3 very large grass plants in it. And then the picture on the right's got a bunch of small grass plants. So imagine you sampling in a plot. Those plots are the same size and you've got those 2 scenarios. So in which plot, 1 or 2, is there actually more grass? It actually kind of depends on what you mean by more or abundant. So let me just get back to there. So in the one on the left, A, if you're looking at frequency, in other words, the number of grass plants, then obviously that's got less than B. But if you were looking at the cover, or how much of the ground is covered by grass, which is basal cover, then B would have more than A. If you're looking at the actual mass, like if you had to clip that grass and weigh it, like gather up all the grass in each plot, then probably about the same. So that's just an illustration of the difficulties in measuring abundance, and you should be aware that it's done in different ways. As long as it's done consistently from year to year, wherever you are, you're going to get an idea. But if you're comparing between different areas done in different ways, you have to be careful. Okay. All right, so the different ways to measure abundance. I mentioned frequency. That's where you would just count the number of individuals within an area, a plot, or if it's a small plot, it's called a quadrat. Or you could look at the cover, and basal cover is how much of the ground has actually got grass or other plant coming out of it. So like where the roots go in. Or aerial cover, which is just the coverage, including the leaves and the stems. So if you're looking down, how much of the ground is covered by the grass plant in any way at all. Often Relative abundance is used instead of absolute abundance. So those measures I've just mentioned are absolute measures, but relative abundance is when you say how much of species A is there compared to species B, for example. And then another very important measure is the biomass one. So how much is there by weight? And for that, for grasses, you would normally either clip them, like I'm showing there a little quadrant in the top left photo, where we go and actually cut all the grass and put it in a bag, put it in a drying oven and weigh it. And so we have a very sort of exact measure of exactly how much grass is within that area. Often that's done in an indirect way with the disc pasture meter. So that's the little instrument shown on the right. This was developed about 50 years ago, and it's quite widely used in South Africa. And here you simply drop a circular aluminium disc onto the grass layer, and the more grass there is, the higher it'll come to rest. If you've got no grass, it'll drop all the way to the ground. If you've got a lot of grass, it'll stop like halfway down the pole, and then you just measure on there how high it is above the ground, and then you can relate that to biomass. So it's a very quick and easy method, although it doesn't work well unless you have quite a lot of grass in your system. This is, of course, for grass. What happens if you're doing trees? Then you can't cut down a whole tree and weigh it. It's not practical if you want biomass, but you can do canopy measurements, where it gives you an indirect measure of how much of that tree there is. And that's particularly useful for working out how many leaves there are, because that's obviously something of most interest when you're looking at carrying capacity and browsers. There's that picture bottom right shows the measurements you have to take for the Beckvol calculations. So Beckvol is a commonly used method that was worked out by some South African rangeland ecologists a couple of decades ago. It's basically a system of measurements that can then give you an estimate of the biomass of the whole tree, or the amount of browse material. Okay, just a note about relative abundance, which I just mentioned. If you're going to use relative abundance, you need to be quite careful, because it only really works well when the total number of plants is not changing. So to illustrate that, here I've got an example. Say in year 1 on the left, you've got some sampling area, a plot there, and you've got 5 of one species, the green species, and 5 of another. So then your green species makes up 50% of the total. So its relative abundance in that simple system is 50%. And then in year 2, maybe it's a good year, there's some rainfall and the green grasses increase. And now there's more of them than the red ones. And so they've gone up to 80%. So there we see a nice change. And that would be an accurate reflection of what's going on in the system. But if something like this happens, where in year 2, there's been a die-off of grasses, now you can see that in year 1, it was the same situation, but in year 2, there's been a loss of both types of grasses. And so we still have a relative abundance of 50%, because the green one is still half of what's there, but there's obviously much less of it. So the vegetation has changed significantly, but the relative abundance measure has not picked that up. So just remember that with relative abundance, it's not where your total number of plants is changing from year to year. Okay, and then in addition to your actual vegetation, it's very useful to measure a few other things that are going to help you interpret the data and understand why your vegetation is changing. And for most ecosystems, in Africa at least, climate and particularly rainfall is really important in this regard. A lot of our protected areas are in arid or semi-arid areas, and rainfall is a big driver of changes in vegetation. So you really need to know how much rainfall you had in the preceding year to understand vegetation change. Some of our cooler systems, temperature would also be very useful. And then herbivores, of course, are obviously a big driver in many systems. So it's really useful to have good data on how many grazers and browsers were in the system for the year. And in some cases, fire, if it's a fire-prone system. So rainfall, even if it's just a manual gauge, like I've shown there, is really critical. The other data sometimes beyond your control, but many reserves do game counts from helicopters or other means, and it's really important to get a hold of that data. For fire mapping, it's in smaller places, it's often done on foot or vehicle, just manually mapping it out. But for larger areas, there are some very nice remote sensing satellite-based tools available. So for example, this is from a website called Firms. which is provided by NASA in the USA, and it's basically it's a kind of a Google Earth specifically for fires. So you can go on there, zoom into anywhere in the world, and it'll show you where it detected fires over the last few days or up to the last month. So this is showing just a small central part of the Kruger National Park, and all the little red dots are areas that burnt in the past month. So that's a great tool for managing large areas. Okay, so once you decided what variables you need to actually measure, you need a system to go and measure them, because you're not going to measure them across every square meter of your whole reserve. So normally you would randomly assign sampling points within your homogeneous vegetation unit. It doesn't have to be random, but that's generally the best for statistical reasons. And then you would sample either within a plot or along a line. So plot-based methods are almost always used for woody vegetation, trees or shrubs, but also sometimes for your herbaceous stuff, your trees and your flowering plants. And it could be a large plot, like 20 by 20 meters, or it could be a quadrat, which is a very small plot, say 1 by 1 meter or half a meter by half a meter. Alternatively, you have point-based methods, which are 1D, so you walk along a line. Sometimes you can lay out a measuring tape, sometimes you just step it out. There are some other interesting methods that have been developed in the past. They're not used as much, like the levee bridge, or what I've shown on the right there is a wheel. You walk that along, and every time a spoke of the wheel hits the ground, that's your sampling point. They work well in certain systems, but the most commonly used is simply a tape measure laid out in a straight line. There's also the point-centered quadrat method for trees, but I'm not going to go too much into detail in that because of time. The last point is repeat photography. So you wouldn't normally use repeat photography for your actual analysis because it's very difficult to quantify and apply statistics, but it's really useful just for, firstly, reminding yourself where the transect is and what it looks like. And secondly, when you come to interpret your data, having photographs really helps you understand. And if you're getting strange numbers, you can look at the photo and try and work out if you made a mistake or if something actually happened. So this is just an example of a line transect and the change in grass biomass from one year to the next at about the same time of the year. One of the most commonly methods used is the nearest plant method in South Africa anyway, for the vegetation, the sort of felt condition assessment that's widely used in game reserves and a lot of national parks. And it involves laying out a transect, a line, and every meter, the distance to the nearest plant is measured. And in some cases, the size of that is measured. So if you've got a very dense grass layer, it's going to look something like that. And your distances to grass are all going to be short. And your tuft size measurements are all going to be big. If it's in poor condition, it's going to look something like that with big distances and small tufts. And then those values are used to create some sort of index about the overall felt condition. I think our next speaker is going to speak more on this, so I'm not going to spend too much time on it. There's quite a lot in the scientific literature about different methods, different sampling designs, testing them in the same environments. So there's a lot of background reading that can be done on this. And that really helps as well to decide what's the appropriate method for your particular environment. Okay, functional grouping briefly. So I did mention that you're going to need different methods for trees versus grasses. So normally your vegetation, you would normally split it into your woody layer, which is trees and shrubs, and your herbaceous layer, which is grasses, as well as the little flowering plants growing amongst the grasses. And then on the woody stuff, you might be interested between tall trees and shrubs. And on the herbaceous side, you're normally interested in grasses versus forbs. You would group those separately. And then, of course, grasses are often grouped further into further divisions because they vary so much in terms of their value for grazers. So one grouping for grasses, the most simple, is palatable versus unpalatable, which is obviously used a lot in agriculture, not so much in conservation. But there are also other functional groups. There's the old idea of succession, and that you have different grass species that colonize first in a disturbed or degraded areas, and then they give way to more specialized grasses, and then they give way to climax grasses, which live in undisturbed areas. So you might see that type of functional grouping. It's not used that much these days. In South Africa, the functional grouping based on response to grazing is widely used. where grasses are categorized as either decreases, in other words, they become less abundant when there's heavy grazing, or increases, increaser 1, 2, and 3, and those increase either when there's too much grazing or too little grazing. Another very basic one is perennial versus annual. So grasses that live more than a year versus those that die every year and have to reestablish from seed, which is more important in your drier systems. And then the grazing value as well. It's very common to assign a grazing value to grasses and then categorize them or group them according to that. Okay, just briefly a bit about grass identification, because it's obviously so important in doing vegetation assessments in many parts of South Africa, in protected areas elsewhere. In South Africa, we're lucky to have some really nice field guides. The one on the left, Guide to Grasses of Southern Africa by Fritz van Oudtshoorn, is a great book. It doesn't have all the grasses, but it's got a lot of them, and it's really useful if you're going to be doing some sort of standard veld condition stuff. There's also some more regional ones that I've shown in the middle, some particularly, for example, the Kruger National Park or Botswana, or the northern parts of South Africa. And then on the right is kind of the Bible of grass identification, the Grasses of Southern Africa. It's an old book, but it is very comprehensive. It contains all the species and it contains all the details that you would need to identify each species, but it is very technical and you really have to be a proper botanist and understand the anatomy and morphology of grass plants to use it properly. And you're probably going to need a microscope too. Okay, so just briefly identifying grasses. The flowers or inflorescences are the key part or the most useful part, but sometimes the structure of the grass plant overall morphology is important. So there you need to know the basic parts of a grass. The leaves are split between blades and sheaths, and where the sheath joins the blade, there's a thing called a ligule, and that's often very useful in identifying a grass because the ligules can have different shapes and forms. The grass might produce stolons, which are sort of sideways stems that grow along the ground, or rhizomes underground, and that's useful to identify. The tillers, so the tillers are the upright stems. They might be branched, or they might have colors at their nodes, so that can be a useful feature. And then, of course, the inflorescence, the flower. And this is where it gets tricky because grass flowers are not simple like most other flowers. They don't just have petals and sepals. They are actually sort of more evolved, and you have grass structures flower structures embedded in other flower structures. A lot of the original flowering plants change to become bracts or other structures that hold flowers within flowers, essentially. So a grass flower is actually called a floret, and those are the parts you can see on the screen there. And sometimes when species are quite similar in other regards, you've got to get down to this level to identify them. So often the awn, it's a little pointy thing sticking off a bract of the floret, is often a useful thing, but it can be sort of the hairiness of the palea or the shape of the lemma or something that actually distinguishes one species from another. Fortunately, for most veld condition assessments, you shouldn't need to get down to that level. The arrangement of the florets on the stem is actually more useful, and you can identify most grasses using this without having to get right into the detailed structure of that floret. So the florets can be are normally clumped together in a grass. So they might be together in a spikelet, which is a whole lot of florets close together, and that's shown in the top left. Or there might be a little stalk on those spikelets, which is then called a raceme. On those racemes might be arranged in fingers, which is called digitate in the bottom left, or they might be all spread out in a panicle. And in some cases, they have an extra structure called a spathe growing around them, which can be quite useful. So just as an example, some common grasses from South Africa fall into these sort of different categories. So the Eragrostis species generally have spikes or racemes, and quite characteristic looking, with a whole bunch of florets grouped together, often in an oval or heart shape. Eurocloa masambacensis is a common grass from bushveld areas, and that's got these straight spikes sticking out at angles, whereas Digitaria eriantha has all the spikes joined to a central point, also a very common grass. And then Thermida triandra has spathes It's a very characteristic looking flower. So Themida and all the Hyperinias often have those, which is quite useful to get them at least to that level. Okay, so to end off, what do you do with all the data once you've got it? You need to do a trend analysis, but exactly what variables are you going to use for your analysis? Sometimes you would just use the exact thing you measured. So maybe you're interested specifically in decrease the grasses if you're running a game ranch, and then you would just measure the abundance of those, check how the abundance of those has changed over time. But often you want a bigger, you're interested in the whole grass community or herbaceous community, and so you combine different species into a single value, an index, which makes it much easier to see what's going on over time. And there's a whole range of indices that have been developed in South Africa. In Africa, the felt condition score is a commonly used one. There's also the ecological index method. And there's a range of others that I don't have time to get into, but just be aware they're there and there's literature on them. And they're normally used to condense all that information into a single value, a score or an index, which is then useful for trend analysis. Sometimes they also use to estimate carrying capacity. So various scientists have developed sort of estimates of how much wildlife an area could support if it's got a felt condition score of X or Y. Okay, and then of course predictive variables come into play when you're trying to understand why you've seen changes over time. So for example, here's a felt condition score index that I've calculated for one of our sites where we've been monitoring since 2014. And this is really just based on the abundance of perennial grasses because it's quite a dry, heavily grazed site, and perennial grasses are key. And we see that it was doing okay, and then there was a large decline from 2016 to 2020, and then it kind of bounced back. So what caused that? Was there some change in grazing? Actually, no, it was pretty much just a drought. So when we look at rainfall for that site, and that's shown in the black bars underneath, we were having good rainfall until 2015, and then we had a really severe drought. And the condition declined, but fortunately, when the rain came back, it bounced back. So this was all under conditions of more or less the same grazing pressure. All right, so I'm going to end off there. Just to summarize, vegetation assessments are unfortunately not simple. They are complicated because there are a range of different vegetation types that require different methods. There's many different variables that can be measured. There's very different sampling designs that need to be applied depending on your variables and your vegetation type. And there's a bunch of indices that can be derived. But what's important, regardless of how you're doing them and where you're doing them, is that you firstly have management objectives and monitoring objectives established up front to guide your selection of methods, and that you apply those 5 principles that I mentioned before to ensure that you get an unbiased and fairly accurate measure and that it feeds back into management. All right, thank you for your time. I'm going to end there.
Thank you very much, Tony. Thank you. That gives us a really fascinating and solid foundation to vegetation surveying, and I think incredibly valuable to all the students in the audience. And I thank you for this. Thank you very much. Given limited time, if the students or any of the audience would like to ask questions to Tony, please type them in the chat. And Tony, if that's possible for you to stay on a little bit and answer those questions, that would be wonderful.